So it's a pleasure to be here today. As mentioned, my interest is the microbiome. And so I will have to tell you from the beginning, I don't have all the answers regarding prevention and treatment, but I would like to present my work on the microbiome and put into that context the topic of probiotics, which I think has come up quite a bit. To sort of share a little bit about how I'm thinking about these issues. So I'll just jump right in with some background to kind of get us all on the same page. I know these are things that we're all familiar with, but I think as we go through this, you'll see how this context plays in. So as we all know, necrotizing enterocolitis is an inflammatory bowel necrosis that primarily afflicts premature infants after the initiation of enteral feeding. We know that the majority of patients are born preterm. In the US, our incidence is about 10% of babies born with a birth weight less than 1,500 grams. Now, the textbooks say that NEC classically appears between 7 and 14 days of life, but we all know that susceptibility is inversely related to gestational age. So the more immature you are, the more likely you are to have this disease and the longer window in which you're susceptible. So this really suggests that there's a developmental component to this disease, and there is no race or sex predilection. The pathognomonic features pneumatosis intestinalis, which is air tracking in the bowel wall, and this represents bacterial fermentation of intraluminal substrates. We don't have specific therapy because we don't yet know specifically what causes this disease. What we do provide is supportive care. So we put the bowel at rest, cessation of enteral feeds, nasogastric suction. We support with fluids and antibiotics and parenteral nutrition. And then there's surgery if there's a specific indication like pneumoperitoneum, peritonitis, or intestinal obstruction. But our outcomes remain poor. Mortality between 30 and 40%. Particularly for those who require surgery, risk of stricture, short gut, poor nutrition, parenteral nutrition-induced cholestasis, and then certainly risk for poor neurodevelopmental outcomes. So for babies who have severe NEC, those who require surgery, there's increased risk of cerebral palsy, a mental developmental index less than 70, physical developmental index less than 70, any neurodevelopmental impairment. Surgical NEC is associated with increased risk of periventricular leukomalacia and decreased growth in all parameters. So you can see from our non-specific therapy and the remaining poor outcomes that there's still a lot we don't know about this disease. What we do know is that there's specific risk factors. So prematurity is the biggest risk factor, along with bacterial colonization, enteral feeding, and hypoxia or altered intestinal blood flow. And I'm going to focus my talk today on prematurity and bacterial colonization. And for a long time in neonatology, I think we've thought about bacteria as the enemy. So everything is in the context of pathogens and how do we identify them and eradicate them. Just to remind all of you, when we're thinking about pathogenesis, if a microbe is thought to really cause a disease, it's supposed to fulfill Koch's postulates, which is that a microbe suspected of causing a certain disease must be found in every case of that disease. The suspected microbe must be isolated and grown in the laboratory outside of the host. When this suspected pathogen is introduced to a healthy susceptible host, that host must develop the disease, and that same pathogen must then again be obtained and re-isolated from that experimental host. So clearly we do not know the pathogen that causes NEC. On the other hand, there's this idea of commensal organisms, beneficial organisms. So Elie Menchnikov in 1882 discovered phagocytes, but in 1907 was one of the first to really suggest the possibility of colonizing the gut with beneficial flora. And he is credited with the introduction of the concept of probiotics. So now we have this tension between maybe good and evil. You have good microbes, you have bad microbes. There's actually a third way of looking at microbes, and that is how we think about the microbiome. And this is the community of all the microbes in and on the human body. There are about equal numbers of bacterial and human cells. So bacterial cells really form a significant population in terms of genetic content, metabolic contribution, and it really is important to recognize that human health depends on microbiota health. And if this is true, we then really need to understand the microbial community perspective. So the microbiome is an ecosystem, just like any other ecosystem, a forest or a pond. The goal is to maintain equilibrium. And in a healthy ecosystem, there is high diversity of species, there's balance, functional redundancy, and therefore resistance to disease. But in a sick ecosystem, there's low diversity of species, imbalance, functional disability, and susceptibility to disease. So when we think about the microbiome and microbes, I just want to put this up there as an illustration of community dynamics. So let's imagine 3 organisms, A, B, and C. They're not pathogens, they're not commensal. We're not talking about them as good or evil, just 3 organisms. And let's just imagine that A produces something that B needs so that B can grow. And let's imagine that B and C share a substrate. And so by the increase in B, there's increased competition for this substrate. And so there's less of C. And let's assume that this is what balance looks like. Maybe increased numbers of A and B and decreased numbers of C. What happens if we perturb this community? Let's say we introduce antibiotics and we take out A. So now A is no longer producing the substance that B needs and B cannot grow. B no longer competes for the substrate, and now C is a greater player in this community than previously. So you can imagine how C may not be labeled as a pathogen, but could have now a different effect on the host. Or let's imagine the situation where there's an altered diet. If you remove the substrate that B and C needs, so that these organisms now disappear, the substance that A produces is now present without B to consume it. And this substance could now affect the host. And you can imagine now in this scenario, A may not be a pathogen, but could cause untoward effects on the host. Just a few more of them. So this situation is called dysbiosis, when there's lack of diversity, loss of keystone species, and overgrowth of opportunistic pathogens. And this is a situation that leads to disease. And it was hoped that by thinking about the microbiome in this way, we could better understand the role of the microbiome or bacteria in necrotizing enterocolitis. So this is where I started my work. And I asked the question, can microbiome analysis be used to identify the bacteria associated with NEC? And for this initial study, I looked at 20 patients, 10 with NEC and 10 control patients. And I happened to have 4 sets of twins where 1 baby had NEC and 1 did not. We looked at fecal samples from these babies just prior to the onset of NEC. So a sample was taken, we had been collecting longitudinal samples and we selected the sample just prior to disease onset in the babies who went on to develop NEC. And we chose a matched sample from control infants matched by gestational age, day of life, type of feeding, and mode of delivery. We analyzed the samples by 16S ribosomal RNA sequencing. This is a way of specifically looking at bacteria. We analyze a highly conserved sequence that's specific to bacteria and then compare those results to databases so that we can identify what microbes are present. Just looking at our initial data, we found that the NEC patients had lower diversity. So diversity is a comment about how many different organisms are there. And as mentioned previously, diversity is associated with a healthier community. So the control patients had higher diversity than the NEC patients. And then when we looked at the phylum level, the control patients only had about 40% of proteobacteria. This includes a lot of gram-negative organisms, and the balance was other phyla. Whereas the NEC patients had an increase in proteobacteria to over 90% of the community, with a very minor percentage being other phyla. One thing to note is that actually both of these percentages would be considered very high. In adult microbiome communities, proteobacteria should be less than 10% of the community. So even in the control patients, there was more proteobacteria than you would see in adults. We then looked at individual patients. So here, the top panel is babies with NEC, and the bottom panel is the control infants. The first 4 are the twins, and you can see the pairing for N1 and C1, and likewise to the N1, N4, and C4. Two things to notice here. First of all, there's very few bars. What's labeled is the dominant genus, but you can see there's very few bars. These are very simple communities. An adult community should have hundreds of organisms, and we have just a handful for each patient. The other thing to notice is that for the neck patients, there was no single organism that was associated with disease. However, each patient had a bloom to more than 50% of their community of a specific gamma proteobacteria. So all of these organisms, while they're different, they are all gamma proteobacteria. We did not see this in the control patients. They might have had a dominant organism in their community, but it was not a gamma proteobacteria, except for these two patients. So these two patients were chosen as controls initially based on their clinical presentation, but they did have more than 50% of Escherichia, which is a gamma proteobacteria, and both of these patients went on to develop NEC in the next 2 weeks. We then specifically looked at twin pairs. So when we matched C1 to N1, so we matched each patient with their specific twin, we saw that the control twins had decrease in proteobacteria compared to the NEC twin, increase in diversity compared to the NEC twin, and that the control patients separated from the NEC patients by disease state. They did not separate out by their twin status, and that wasn't necessarily predicted. When we looked at the complete cohort, we also saw separation. So what this is a PCA plot of a unifrac analysis. So each dot is a representation of a complete community in three-dimensional space, although this is a two-dimensional graph, obviously. And the closer the dots are together, the more similar the communities are to each other. And if there's separation, they're more distinct from each other. And you can see that the blue squares of the control patients do separate from the red dots of the NEC patients, except for these two. Again, these are the same two patients that we had chosen as controls who went on to develop NEC later. They actually clustered with the NEC patients. So this data told us two things. First of all, there isn't a specific pathogen associated with NEC. However, there is a microbial community associated with NEC, and it changes before NEC onset. So this led to another question. When do microbiome patterns change, and is it at a time point early enough to intervene? And for these studies, we looked again at our patients with and without NEC, and this time we analyzed the weekly fecal samples over time. And these pie charts are another plot where the samples that are closer in space represent those that are more similar to each other. And what you can see is that the initial samples of patients were similar to each other, but those who went on to develop NEC followed one trajectory, and those who remained healthy followed a different trajectory. And at this point in my research, I had a choice to make. I could either continue to follow the babies that went on to develop NEC, or I could follow those who remained healthy. And I actually chose to follow the ones who remained healthy because if you think about those risk factors, actually all of our babies are preterm and colonized with bacteria and are enterally fed and have altered intestinal blood flow. There must be something about those who don't go on to develop NEC that is perhaps protective. So then I analyzed the microbiome over time of just the babies that did not go on to develop NEC. And I compared this analysis to samples from a healthy full-term breastfed infant cared for at home. So this would be considered the gold standard of a healthy baby. You can see that for the full-term infant, weekly samples collected over about 8 weeks of life cluster pretty closely together. So we chose 8 weeks as sort of an average length of stay for a preterm infant. In that time frame, the infant cared for at home, the samples are pretty similar to each other. However, what we found was that there was a temporal progression of the preterm infant microbiome with a clustering around 2 weeks of life, another cluster around 3 to 5 weeks of life. And in these open white squares, it's only after 6 weeks of life that they begin to converge on the pattern of the full-term breastfed infant. So this suggests that there may be a developmental progression to the microbiome of the preterm infant that the full-term infant doesn't undergo. And this may make sense because the full-term infant doesn't have the same developmental hurdles that a preterm infant does. And this is consistent with what we know, that prematurity is not a disease state, prematurity is a developmental state. The preterm infant undergoes sort of a mistimed change in environment. So rather than completing development in the intrauterine environment, which is a relatively sterile environment where the placenta does all the work of oxygenation and providing nutrition, the preterm infant develops in the neonatal intensive care unit. And in this environment, host development coincides with microbiome development. And I wondered what role the microbiome could have in maturation. So we know there's many aspects of the preterm gut that are immature and may predispose to NEC. We know there's immaturity of cell surface glycoconjugates, which alters what microbes adhere to the intestinal surface. We know there's alteration in many aspects of innate immunity, decreased levels of gastric acid, decreased intestinal motility, decreased levels of secretory IgA, decreased levels of bacteriostatic proteins, intestinal trefoil factor, and human defensins. There's immaturity in intestinal mucus, which is intended to confine bacteria to the luminal space. Differences in terms of density and incorporation of secretory IgA. There's supposed to be tight junctions between cells, again, to limit crossing of bacteria, but this barrier is actually leaky in preterm infants, and this is thought to be a holdover from intrauterine life, where the amniotic fluid has large macromolecules that are important for development and important to allow them to cross. There's decreased numbers of B and T lymphocytes in preterm infants, and for the enterocyte itself, we know that there is increased NF-kappa B activation, which is associated with an increased inflammatory response to microbes, actually both pathogens and commensals. So I wonder, does the microbiome have a role in intestinal development? The problem, though, is it's actually easier to define a disease than sort of define development or a healthy outcome. So we needed something that we could measure to answer this question. And we chose to look at growth. So definitely studies have shown that babies who gain weight more rapidly while in the neonatal intensive care unit have improvement in long-term outcomes. Growth is sort of a summary metric that suggests adequate nutrition, resilience, resistance to infection. So this is what we chose to measure to look at the role of the microbiome in development. And for these studies, we used a gnotobiotic mouse model. So gnotobiotic just means known biota. And what we did was we studied germ-free mice and gave them specific microbiota. So the gnotobiotic mouse model is used quite a bit in microbiome research, but I would argue it's particularly relevant to preterm infants, where there's a specific time point after which it is the normal situation where colonization rapidly increases. There's a prolonged period of observation for our preterm infants, so a prolonged hospital course, a regulated diet, simple communities, and known follow-up. So for these studies, we took samples, fecal samples that contain the microbiome from infants with two different growth rates, high growth and low growth. And we all know that there's babies in the nursery. It doesn't matter how many calories you pour into them. It just seems that they can't grow. It seems there must be some other factor. We sequence samples from these babies, and going forward, H will signify high growth, L will signify low growth. And we took samples from the first 2 weeks of life, that first cluster that seemed to be significant, thinking that these initial microbes that encounter the naive intestine may have the greatest opportunity to have a significant effect on fundamental pathways. So 6 days old and 12 days old, both within the first 2 weeks of life. You can see in black that there were many common organisms. In red and blue are the organisms that were different. And you can see again, there actually weren't very many organisms. So these are simple communities. We took fecal lysates from the babies, transphonated them or fed them by gavage to germ-free pregnant dams. And then the pups that were born, the litters acquired the microbiome of interest naturally from their mother through delivery and contact with the mother, which is sort of what happens to babies as well. We measured daily weight until weaning, and what we found was that high growth baby led to high growth pup, and low growth baby led to low growth pup. So now we have a model where we were able to recapitulate our clinical phenotype, but could also look closely at tissue. These pie charts just demonstrate that we were successful in intertransformation. SPF is specific pathogen-free mouse pup. This is standard mouse colonization. You can see yellow and blue. This is Bacteroidetes and Firmicutes. But you can see that all of our transphonated mice, either the low growth dam, the low growth pup, the high growth dam, or the high growth pup, all have this purple color. This is proteobacteria, and similar to our babies, there's a significant percentage of proteobacteria that is not supposed to be in the mice. So this shows that we were successful in our transformation. And then we looked at the tissue. So as a measure of growth, we looked at intestinal length. This would be associated with nutrient absorption. You can see in the high growth, there was a difference in length compared to low growth. Over here, this pink staining is Ki67. It stains for cells that are proliferating, and you can see there's increased proliferation in the high growth compared to the low growth. Then we looked at markers of innate immune defense that we know are immature and preterm infants and are thought to be associated with NEC. So we looked at numbers of goblet cells. These are associated with producing mucus. Many more goblet cells in high growth than low growth, and it was statistically significantly different. There were differences in Paneth cells. So Paneth cells produce defensins, increased numbers in the high growth compared to low growth, and this was statistically significant. We looked at differences in tight junctions. So these were, again, the sealing between intestinal epithelial cells that are important for confining bacteria to the lumen and not allowing them to cross the intestinal barrier. We looked at two proteins, occludin and ZO1. You can see in the low growth, this is overall, and this is a blown up picture, they're these bright red dots. They're just scattered in the low growth, but you can see this nice continuous pattern in the high growth that suggests an intact continuous barrier for both of the tight junction proteins. And then we looked at inflammation. So you don't need to know what any of the words are, except this is low growth and high growth. What you should be able to see is that there's a lot of orange in the low growth, and orange signifies upregulation of genes associated with inflammation. So these are depictions of inflammatory response networks, and you can see that in low growth, there's much more upregulation of inflammation compared to high growth. And this was just in response to a difference in what we gave the mice in terms of microbiome. There was no secondary inflammatory stimulus. When we looked at specific inflammatory cytokines, blue is low growth, the purple color is high growth. You can see that for all the ones that we measured, there was increased production at these are samples from the ileum in the intestine, increased production of inflammatory cytokines just associated with the microbiome. And then we looked at NF-kappa B activation. So NF-kappa B is a transcription factor important for transcribing genes associated with inflammation in the immune system. You can see in low growth, there's much more activation of NF-kappa B, again, just in response to the microbiome compared to the high growth. And the other striking thing is that high growth is actually lower than SPF. So SPF is normal mouse colonization, H was actually lower. And so what this suggested to us is that the microbiome could do two things. The microbiome could increase your risk of having inflammation, or it could actually decrease your risk of having inflammation. And this would be associated with a maturation in your intestinal phenotype. So this again suggests that the microbiome could do beneficial things or harmful things. So this brings into question, how do you ensure then a beneficial microbiome? How do you change the microbiome? And I think this is where a lot of the interest in probiotics has come from, just this desire to improve the microbiome. So probiotics are living microorganisms in food and dietary supplements, which have beneficial health effects beyond their inherent nutritive value. But there are specific requirements. There should be benefit without toxic effects. The organism should remain viable during storage and use. They should retain the ability to colonize the gut, be capable of surviving in the gut, and have well understood properties. And the truth is, although probiotics have been used in clinical trials, we don't have a probiotic that meets these requirements. But the interest in probiotics has remained. There's several studies that have been done. I did not include the ProPrem trial here, but these are representative. So each of the trials have shown a decrease in the incidence of NEC. However, there were differences in the probiotics that were used in these different studies. So it's not really clear what a recommendation would be. I think another concern with probiotics is that since we don't have a biomarker for who is most likely to develop NEC, probiotics have to be given to all preterm infants in order to decrease the incidence of NEC in some. We can't predict who's at higher risk. Therefore, there is a significant percentage of patients who would receive probiotics, who would be treated, but not at risk for NEC, because the incidence of NEC across the whole population is somewhat low, even if you pick a higher number like 10%. The ProPrem trial had in their control group only 4%. So there's a lot of babies who would be treated but not at risk for NEC, and that raises safety concerns. In addition, there are still open questions about what's the optimal probiotic organism, who do you give it to, dose, timing, length of treatment, mechanism of action is not understood, and there is not standardization of preparations. And there is a real infection risk. So there have been studies that have been published of bacteremia in preterm infants on probiotic therapy. There was a fatal case of gastrointestinal mucormycosis in an infant following use of ABC-Dophilus, which was used in a lot of the trials. And they did find that the probiotic itself was actually contaminated, and there are not specific guidelines for manufacture of probiotics. So the question is, should we give probiotics to a patient who is never going to get to develop NEC? And at this point, if I was in a smaller audience, I would ask, is there a time where we give an agent to a large population of people, of infants, to prevent disease in a few, and an intervention that we know has significant effects long-term and on the immune system? And I think the only time we do that is really with regards to vaccination. But what is the difference in the type of data that we have for vaccines versus probiotics? So I've heard it say, well, there's so much more data on probiotics compared to surfactant or cooling for HIE, yet we are very comfortable with surfactant and cooling and much less confident about probiotics. But if you instead compare the data we have for probiotics to vaccine safety, for vaccines, safety and effectiveness is under constant study. They have to be extraordinarily safe. Safety testing begins as soon as a new vaccine is contemplated, continues until it is approved by the FDA, monitored indefinitely. And this has not been the case for probiotics in preterm infants. So then the question becomes, is there a different way to modify the microbiome? Is there a different way to end up with a beneficial microbiome besides just giving probiotics? And there are other ways. So this is a study on the role of human milk in extremely low birth weight infants. So in this study that looked at human milk intake in over 1,000 ELBW infants over several centers, when they looked at NEC or death between 14 days and discharge, they found that the hazard of NEC or death was decreased by 13% for every 100 mLs per kilo increase in human milk an infant received in first 14 days of life. Increasing the amount of human milk was associated with increased survival time free of NEC, and increasing proportion of human milk to total intake was also associated with decreased relative risk of NEC. So they concluded that a dose-related association of human milk feeding with a reduction of risk of NEC or death after the first 2 weeks of life exists among extremely preterm infants. Another way we could improve the microbiome is perhaps by limiting our use of antibiotics. So this is another study that looked at many infants, over 5,000 admitted to 19 centers, looking at NEC or death, NEC alone or death alone. What this study found was that many infants receive prolonged empirical therapy, which is defined as empirical antibiotic therapy for more than 5 days. This prolonged therapy was associated with increased odds of NEC or death. In fact, each empirical treatment day was associated with an increased risk. So these authors suggest that prolonged initial empirical antibiotic therapy may be associated with increased risk of neck or death and should be used with caution, and speculated that choice of antibiotic duration is likely to affect the colonization of the neonatal intestine, which in animal models seems associated with the physiologic development of the intestine, immunologic development, absorbance of nutrients, and alterations and variation in any or all of these areas could increase the risk of neck. So if you think now about modification of the early microbiome and neck, we actually have three therapies. You could give probiotics, you could decrease number of days of antibiotics, or you could give actually any breast milk. All of these decrease the incidence of neck. Now, these two studies were from 2009. This is from the 2014 Cochrane review. And you can see here that the incidence of neck in the control population is already much lower than previously. So there's also other things that we are doing in our overall neonatal care that decreases the risk of neck, separate from probiotics. And I put this up here as a caution. So these are multiple strains of a single organism, Lactobacillus reuteri, and this shows all of the cytokine profiles associated with different strains. And the main takeaway here is that they are all different. There's some decrease, there's some increase of the same cytokine. So consideration of host origin and functional differences at the subspecies level may have major impacts on probiotic strain selection and considerations of microbial ecology in mammalian species. So, it's very important when we think about probiotics to consider strain, and we are not to the point where we know strain level effects of these organisms. So what I would say that the facts are the microbiome is important in health and disease, but the term probiotics encompasses many different organisms, and we don't know what the probiotics are doing to the preterm infant microbiome. Under current protocols, the majority of preterm infants that would receive probiotics were never going to develop neck, and we can't say that probiotics are without harm, particularly for the infants who are not going to develop neck, and the studies have not followed these babies long term. And we have been here before. Things like oxygen and dexamethasone were used readily, and good things can be harmful at key developmental stages. I think we've all learned some caution. So I would suggest that rather than messing with the preterm infant microbiome with probiotics, we could think about protecting the preterm infant microbiome and recognize that probiotics have effects on the microbiome that we don't understand specifically with regard to impact on the overall microbial community. But what we can do instead is evaluate therapeutic options in light of effect on the microbiome, specifically decreasing antibiotic use, promoting exclusive breastfeeding. And these are potentially ways that we can protect the preterm infant, improve the microbiome, and induce intestinal maturation as well. So thank you, that's all I had today.